to study, that is about translation, right? Till here we studied start codon and stop codon. The last point which we studied in the previous in the previous session, that was about start and stop codon. Now we are supposed to start from translation. What is the meaning of translation? Who can tell me? Just general.
Translate to be.
To be what?
Anything.
Huh? Translation mean what?
Translate.
Change what? Huh? Translation. Just in simple English. What is mean of translation? What do you say in Arabic? I'm saying just general. What is the meaning of translation? Not translation mean change of language, like English to Arabic.
Yes.
Not to place. Change of language. Right? Yes. Okay. Translation mean to translate something, right? Translate something, or you can say like, what do you say now? Tarjama. Okay, so translation is the process which will continue in the protein synthesis, right? Now, if you remember till here, we said that on the DNA, what do we have? On the DNA, we have genetic codes. On the DNA, we have genetic codes. Now, their genetic codes are going to be translated into proteins, right? How their genetic codes are going to change into proteins, or we can say into RNA, right? So, change of genetic code into protein, that is called what? Translation. This process is called translation. How it will occur? We have to study it, right? The decoding of messenger RNA messages into protein. If you look to the DNA, right? So the DNA contains all the information. All mean the DNA contains all the information for all the processes in the body. Which type of protein is required, right? In which part of the body, and all these processes, it is present on what? It is stored on the DNA. Now from the DNA, we know that from the DNA, a small short sequence, right? A short sequence or a small portion of the DNA will be translated into the RNA, will be transcribed to the RNA in the first phase. Into what? Into messenger RNA, or we can say premature messenger RNA, which is not mature. So after editing process, it gets mature and it's come to travel from the nucleus to the cytoplasm. Ribosome use the sequence of codon in messenger RNA to assemble amino acid into polypeptide chain. Right? Now, ribosome. What is ribosome? This is a new thing, right? This is a new word. Ribosome use the sequence of codon in messenger RNA to assemble amino acid into polypeptide chain. What is amino acid? What is ribosome? Ribosome is an organelle. Ribosome is what? Ribosome is an organelle which is present in the cytoplasm of the cell. It's an organelle which is present in the cytoplasm of the cell. So ribosome will use the sequence of the codon which is coming from the DNA, right? It will use the codon in the messenger RNA to assemble to collect amino acid into a polypeptide chain. So ribosome will read, right, or will identify the codon which is coming from the DNA. In what? In the messenger RNA. So ribosomes are going to identify the codon in the messenger RNA, and beside this identification, right, beside the identification of the sequence, the ribosome are going to assemble, are going to collect amino acid, right, the concerned amino acid, and put it on and attach it to the polypeptide chain. Polypeptide chain is what? Polypeptide chain is the protein which is going to form, right? The protein which is going to form. So this is not as easy process, right? If you look in the book on page number 368 and 369, so you can see the diagram, right? And you can even see here, right? Now we will go a little bit to the previous lesson, right? Little bit, right? Not too much. Okay, if you look here, this is what? This is messenger RNA, right? This structure is of what? This structure belongs to messenger RNA, right? Now you can see on this structure we have what? On this structure we have A, U, G, U, C, U, U. These are what? What are these things actually? What are these points? You can see here, right? I told you this is messenger RNA, right? From this, now the protein are going to form. It's going to form protein. So here we have A, U, G, U, U, C, A. What are these things? Anyone? Codon. These are codons, right? Or these are amino acid sequence. Codon or amino acid sequence? Codon. These are amino acid sequences, right? These are amino acid sequence, but if I say you are correct, so yes, you are also correct. These are also codons. We are saying that when 3 amino acid comes in a sequence, in one group, we call it codon. When 3 amino acid come in one sequence, we call it what? We called it codon. Like you can see here, this is what? This is the sequence of amino acid, which you can see, right? This is the sequence of this is the sequence of nitrogenous bases, right? AUG. So AUG. This is what? When these three come together, mean it is going to code for what? It is one codon and it is going to code for amino acid. So now who can tell me AUG will code for which protein? Which amino acid? Yes, AUG. AUG is responsible for coding of which protein amino acid? Yes, come on, please. What? For which amino acid it will code? AUG. AUG will be always the first amino acid. What is the first? Mean the starting amino acid. Promoter. AUG. AUG. Methionine. Is it methionine? A. Look to A in the central. In the center, look to A. Connect into U. In the second line. And then into G. What is there? What is
This methionine.
methionine, right? So methionine is the only amino acid which is start amino acid, right? From which the protein synthesis will start. The first amino acid from which amino acid will start in every case, right? Methionine, which will have a code of what? Which will have a code of AUG. AUG, right? And stopping codon we have 3, right? It's given here in the previous lesson we discussed. So AUG will always we will have methionine from which the protein will start. That will be the starting proteins. That's right. AUG is what? AUG is the starting amino acid, which name is methionine. Transfer RNA or tRNA, right? Transfer RNA or tRNA. Each tRNA or transfer RNA molecule carry just one kind of amino acid. Now, the amino acid which we say that ribosomes will identify. Ribosomes will identify the genetic code. And the next process, the next step is that ribosome will assemble, will collect amino acid and will aid on the polypeptide chain, right? So that is what that this transfer RNA that it will take. This is transfer RNA which will take amino acid and which will bring it to the polypeptide chain, right? To the polypeptide chain here we have on this point, right? So the first methionine was there, and let's suppose another should be anyone, right? Anticodon, 3 unpaired bases, which is complementary to messenger RNA code, right? So transfer RNA will have what? Transfer RNA will have 3 anticodons, right? Opposite to that of the one which is present on the transfer RNA. Sorry, on the messenger RNA, right? So transfer RNA will have anticodons, right? Which will be opposite to that of the transfer RNA. Like here, they give you the example of this. Here, you can see this one, right? AAA. So A adenine will always bind to thymine. In the case of the RNA, it should be uracil, right? So AAA to UUU. No, it's not from this one. It's from the previous lesson. Okay? And UUU is what? UUU is. You don't have books. You check U in the subject. Huh? UUU? Phenylalanine or lysine? U. Phenylalanine. Yes. Do we have any other UU? No. So phenylalanine, right? Okay. What is AAA? AAA. No, lysine, right? So it is lysine. So it depends on what? It depends on the codes of the transfer on the messenger RNA, right? We are going to know it from what? We are going to know it from the messenger RNA. Remove your cap. Remove your cap. Remove it. Okay, now the process of translation you can see a little bit more in detail here at this point, right? If you understand it at this point, so mean you understand it completely, right? Now the ribosome, first step, right? What are the steps? We have 6-7 steps in this, right? Just open your mind here completely. Look here, I will ask at the end of this. Everyone need to know. The first step here, it is a ribosome attached to the messenger RNA molecules in the cytoplasm, right? So that is ribosomes which is going to attach into what? Ribosomes which is going to attach into what? To the messenger RNA in the cytoplasm, right? Not in the nucleus, outside the nucleus, in the cytoplasm. The second step, as the ribosome read each codon of the messenger RNA, it direct transfer RNA to bring the specified amino acid into the ribosomes. The function of ribosome, as we know that ribosome are going to read the codon present on the mRNA, right? So the codon which is present on the messenger RNA, it will be read by what? It will be read by the ribosomes, and it will direct message to the transfer RNA to bring the specific amino acid, right? Like for example, the first one we have AUG. So AUG is read by what? AUG is read by the ribosomes, right? And AUG ribosome know that AUG stand for what? AUG code for methionine, which is the start codon. So it will give message to the transfer RNA to take what? To take methionine and bring it here to this point. Right? So when it attach, then this will release the transfer RNA will release back, right? And amino acid will form. Amino acid will be there. This from here, protein will start. The synthesis of protein will start, which is methionine. Okay. The second one at a time, the ribosome then attach each amino acid to the growing chain and break the bonds between the transfer RNA and the amino acid. The next function of the ribosome is to do what? To bring the amino acid and detach the amino acid from what? From the ribosome, from the transfer RNA. So the transfer RNA will be free, it will be removed, right? Now it is the turn for the next one. Now AUG is the first, then we have UUC. What is standing up UUC? Check. UUC. This phenylalanine, right? UUC is code for what? Phenylalanine, right? So phenyl, the ribosome will give message to the transfer RNA to bring what? The second amino acid after methionine, which is phenylalanine, right? So phenylalanine is bring here, it will be attached. Next one we have AAA. So AA stand for lysine, right? It stand for lysine. The message will go back and lysine will come and it will attach at this point, right? So now we have methionine, we have phenylalanine, and we have lysine, right? These three now. So 3 we have already. Now what is remaining? Termination is remaining, right? 3 is enough, but it will not be 3. This is just we are doing example in the 3. It will have more than thousands of amino acids, right? A lot of amino acids will be there. Step 4, the transfer RNA moves into a third binding site from which it exits the ribosomes, right? So it will move and it will exit the ribosomes. The ribosomes move to the third codons where the transfer RNA bring it to amino acid specified by the third codon, right? Like for example, third we have what is the third one we studied? Lysine, right?
Yes.
Lysine was the third. If you look to the direction of the translation, so it will occur in this direction, right? Starting from methionine, right, which is the first one, methionine. Then we have phenylalanine, and the third one we have lysine, right? And now, which one is this? Let's check it, right? The fourth one. The polypeptide chain continued to grow until the ribosome reaches a stopping codon. So it will grow in the same way, right? That ribosome will detect, ribosome will read the codon, it will give message to the transfer RNA to bring the specific amino acid. When it attaches, so it will detach, it will break the bond between what? It will bring the bond between the transfer RNA and between the amino acid, right? So till which time it will continue? Till that time where the stopping codon reach. How many stopping codon we have? 3. Check it. What are it?
The first one is
UGA, UAA, or what? 3 stopping codon we have, right? UGA we have, UGA we have, UAA we have, and UAG we have. So any 3 of them, right? Any 3 of them when comes, so it will do what? On this, the protein synthesis will stop, right? So if you look in this case, we have what? In this case, here we have UGA, right? So it is a stopping codon. So when UGA comes, so mean that it will terminate the protein synthesis, right? Mean it will show that the protein is now complete, right? So we have what? We have here, this is a polypeptide chain. And this polypeptide chain is what? So it is a protein, right?
yes.
It is a protein. It is a protein fragment, you can say. When the ribosome reaches the stopping codon, it releases both the newly formed polypeptide and the mRNA molecules, completing the process of translation. So at this point, the process of translation terminates, mean it's complete. Right? You can check it. If you have any doubt, so you can ask me. Then we have the last point of this lesson, which we will take. Central dogma of molecular biology. Yes, or the molecular base of heredity. Central dogma means main theme, right? Main idea, main theme. What is the central dogma of biology? Central dogma of biology. Main theme of biology. Organization of life we have, right? Cell, tissue, organ, system, and organism. That is what? That is the central dogma of biology. Now, molecular biology is the branch of biology, right? Molecular biology, the one which we are studying, DNA and RNA. This comes in which branch of biology? DNA and RNA comes in which branch of biology? DNA and RNA. It comes in which branch of biology? Yeah. Which branch of biology? Biology have many branches. So which branch we are studying now? Which branch? Molecular biology. I told you it's molecular biology, right? In which we are going to study on molecular level. DNA and RNA is what? Biomolecule, right? It's molecule of life. So in this we are going to study molecular biology. Now information is transferred from DNA to RNA and then into protein, right? DNA, RNA, and then into protein. So it means that the central dogma of molecular biology is what? Related to your lesson, that is DNA, RNA to protein, right? DNA to RNA to protein. That is the central dogma of molecular biology. Mean the main theme that how from the DNA come to RNA, how from the RNA come to protein. That is it, right? Now, if we look to the information is transferred, if you look to the entire information a person have, a plant have, an animal have, it will be present in the DNA, right? But from that information, the function will be carried out. It will be transcribed first to RNA, and then it will be translated to the DNA, right? Exception includes retrovirus, as like we have example HIV, where information is moved from RNA to DNA. We have some exceptions, right? The exception is in the case of what? The exception is in the case of the retrovirus. Those viruses which has genetic material RNA, not DNA, right? Which has genetic information RNA, not DNA. Like in case of the HIV, we have now recently a new virus, which the word is missing, COVID virus, right? Coronavirus. So it also contains RNA virus, right? It also contains some RNA viruses. So in this case, then it is exception, and then the central dogma can be RNA to DNA, then to RNA, then to protein, right? Now gene expression. The way in which DNA, RNA, and proteins are involved in putting genetic information into action in a living cell. What is gene expression? What is gene expression?
The way in which they will have RNA and proteins are involved in vaccines.
Yes, right. Like for example, now our body needs some which specific type of protein of, like for example, histine, we need lignin, right? Protein, any type of protein our body needs. Let's suppose take example of lignin protein, right? So lignin protein is required by the body. The information which is present, which is required for lignin protein, it is present here. It is present in the DNA, in the nucleus, right? So now that short sequence of the lignin protein which is present on the DNA, it will be translated into what? It will be transcribed into messenger RNA, and from messenger RNA it will be transferred into the protein synthesis, right? It will be translated into the protein, and it will form what? It will form protein. So that is what? That is action, right? Here, if you look to this diagram, this is your entire lesson, right? This diagram is your entire lesson. What we studied, right? And the RNA in the second this lesson, right? Which is RNA and protein synthesis, right? So this diagram illustrates all the process. What we studied till now. You can see this is what this point, the first point. You can see this one. This is what? This is DNA, right? First, what we see that how from the DNA RNA are going to form, right? The strain, this is the DNA strain. From this, how the transcription occurs, and based on the transcription, RNA is formed, right? But this process, the formation of messenger RNA, it's occur in which we have RNA editing. We studied here RNA editing. We studied start codon. We have studied splicing, exon, intron. These all we have studied, right? So it's all process. That all process occur in this part, right? Inside the nucleus. This is the boundary of nucleus, right? This is what? This is nuclear cell, a nuclear wall, right? Or nuclear membrane. So after this, what will happen? The messenger RNA is formed, and it will be translated, or it will be, yes, it will be translated out from what? It will be translated out from the nucleus, right? So when it comes out from the nucleus, so the topic today which we study, right? That ribosome will read it, ribosome will give message to transfer RNA to bring the specific amino acid and attach it on the polypeptide chain, right? And when the stopping codon reach, so it will terminate what? It will terminate the protein synthesis, and that will be, it will release messenger RNA, and it will release the protein synthesis. It will release messenger RNA and will release proteins. So that is the topic for today, which is finished. If you have any type of doubt, you can ask me. Which you can see, this is what? This is a portion of polypeptide chain, right? Alanine, arginine, and leucine, 3 amino acid, right? These are 3 amino acid, right? You're supposed first, if it start from this point, so here should be methionine, right? And if it stop on this part, so here should be stopping codon, right? So that If it is a long chain, so it will convert later on to what? Convert later on into protein. What is genetic code? We studied this, right? Okay, one point, the universal genetic code. Let's have a look here. The genetic code is universal. What does it mean? Some organisms show slight variation in amino acid assigned to a particular codon, but the code is always read 3 bases at a time and in the same direction, right? So it will be in all organisms, it will be the same way, right? This diagram we have already explained. So if you are going to get how many protein we have, total number of amino acids, sorry, not protein, amino acid. How many amino acids we have? Amino acid. I told you, it's not written here. No. 20 amino acids we have, right? Total number of amino acids we have 20, right? If you count this, how many are this counted? In figure 13.6, total number of how many we have? Starting from histidine, just take start from histidine and count it. How many protein we have in this? Histidine. Yes.
Right.
25, right? 25 we have, so we have 3 stopping codon, right? So remove 3, 2 from that, and there is also some repetition, right? So total number we have 20.
The tutorial remove 2 arginine.
That we will stop here, right? That's end of the lesson.